So, a very good afternoon to our esteemed speaker, our respected principal sir, my colleagues, and all the participants joining us today. It is indeed a privilege to welcome you all to this international webinar series titled Global Dialogues in Pharmaceutical Sciences, Innovation, Translation, and Impact. which is a remarkable initiative by the School of Pharmaceutical Sciences, City University. This series brings together renowned international experts from diverse specialties like clinical pharmacy, nanomedicine, regulatory sciences, precision medicine, and drug delivery systems to share their insights, innovations, and real world experiences. Now it is my honor and privilege to introduce a highly respected academic leader who will now share insights about our department, Dr. Veer Vikram Sir, the principal and professor at City University. He is distinguished academician and administrator with over 25 years of experience in pharmacy education, research, hospital administration, and institutional leadership. He has made remarkable contributions to the pharmaceutical field through more than 35 research publications, numerous national and international presentations, chaired international conferences, delivered invited lectures in countries like Malaysia, Taiwan, Dubai, and many more. As the principal of City University, Sir has played a pivotal role in strengthening the academic framework, research culture, and global collaborations. His dynamic leadership has guided the university toward excellence in pharmacy education. Now I request Dr. Veer Vikram Sir to present a comprehensive introduction to our department.
So thank you, Dr. Lakveer.
Thank you, sir.
Actually, today is our esteemed speaker, Muhammad Irham, not for me. So good afternoon, Dr. Muhammad Irham, and welcome for this series. Today is the second series. First today was the professor from the USA. So I will just present my PPT. I request to the Dr. Dilpreet, please present the PPT. That is a little bit information about my school. So because you are from very far away from the India, so I want you should be know that about the City University. So I will only show the different research that is only going on in our department, and my team day and night working on this segment. So Doctor, in City University, the different research that is going on, that is, one is our group is old from 1997, and our concept is pre-nursery to PhD. So our leaders is Sardar Charanjit Singh Channi ji, our chancellor of this university, our pro-chancellor Dr. Manbir Singh, and our vice chairperson is Mr. Harpreet Singh. So under the leadership of our Vice Chancellor, Dr. Nitin Tandon, we are doing the research in our department. And our Pro-Vice Chancellor, Dr. C. Ramesh, he's actively participate in BA, BE facilities and preclinical, pharmacological and toxicological and synthetic R&D facilities. And myself, Dr. Veer Vikram, and as per Dr. Lakveer already explained about myself. So, Dr. Mohammed, recently we will visit to the TechU universities, and we have tie up with the TechU universities regarding the lab exchange programs. So, what is the pharmaceutical research in my university? So, in our universities, we have drug delivery and development system. We work on a clinical research. We have pharmacological and pharmacokinetics activities, and we have tie up with the hospitals. So we are working on even though trials also. And whenever we're working on the trials, we have two types. One is preclinical research, another is clinical research. In our preclinical research, we work on drug discovery and preclinical research. Recently, we work with one pharmaceutical company that is DuPont, where we work on antibiotics. That is the first time in my university we convert that antibiotic injectable information to the company for further approvals. So, doctor, in our lab, we have zebrafish lab, we have a Drosophila lab, we have approved animal house where we have rat, rabbit, guinea pig. And in our lab, we have started anthelmatics lab, and very soon we will start silkworm lab in our universities. That is the approved animal house. And we have zebrafish workshop that is in collaboration with the that is alternative to animal ethics. We start that workshop, and in our lab we work on crude drugs to marketed drug. One of our students, he work on the depression antidepressive drug. He procure it and then finalize for research. So on the students work on that is zebrafish. Naturally, if you will do the work, our media persons will interact with us. So all media cover us. So this is our Drosophila lab. We work on Drosophila, that is endocrinologies. And the one leading newspaper, The Tribune, he cover all the segments. So in our pharmaceuticals, different instrumentations are there. In silico drug discovery lab, one of your friend, Dr. Dilpreet, is working on that particular labs. And industrial development project I have discussed with you in collaboration with DuPont companies. We have worked on that segments, and now we have ICHQ8. We have applied for that because injections to tablet conversion is not so easy for us. Nano carrier system drug delivery, and my team is working on that. And we have collaborative research. And definitely, after your lecture, I would like to listen from your side. We, in collaboration, we can also work in your lab, and you can also collaborate with us. Same thing, we have cardiovascular lab. That is our pharmacology students and others, the students from the different hospital. They work with us in silico pharmaceutical lab because we reduce the experiment cost and we have enhanced the prediction accuracies. And one of the professor from the Uganda, he visit our lab, and in collaborative he will interact with us. Few students from the students from Australia, they also work on this zebrafish. And I told you we recently visit Turkey, where we work on the silkworm, and in future we will start this lab in collaborative with EQ universities. So, doctor, we have more than 200 publications, and we have industry collaborations. We have more than 50 published patent in our hand. So these are the strengthening our university, and the hospital people they visit to check the preclinical trial in our department. So this is the little bit information, and this is the our team. They are working day and night, and I'm very happy because our pro vice chancellor sir just joined now, and he's
Yeah.
listening, and he's. I welcome our pro vice chancellor sir C Ramesh sir, and he is too much busy. And whenever I told him, sir, our Muhammad Irham is today is our speaker, he told me I will interact with him few minutes. So welcome,
Yes,
sir.
sir. My hearty welcome to Mohammed sir. Uh, very happy to have you in this seminar, and we are very much happy to listen to you in this area. Then and to very we have been discussing with Veer Vikram, Dr. Vikram, about Center for Digital Health. Sir, we are also planning for digital health, where we want to work on precision medicine, also precision medicine, and then digital therapeutics, which is very highly developing now. So we want your suggestions and support for this, sir. Actually, just I want to put that point to you. And since I am very busy in some other meetings, I'll just interact with you and and then leave from this seminar. You can go ahead, sir. Can you support us in this direction, sir? Please.
Pardon, sir.
Uh, just for Center for Digital Health, I would like to ask Mohammed sir to support us in precision medicine and health, digital health, and all those things. Yeah. Can you listen? Can you hear me, sir? Mohammed, sir.
Yes, bro, we can hear you.
Yeah, yeah,
Yeah,
yeah. We want your support, sir. Thank
of
you,
course.
sir. That,
Thank you
yeah,
so much
yeah.
for the
Thank you, sir.
opportunity.
Thank you. Yeah, thank you, sir. Yeah,
Thank you, Ramesh sir. Now,
yeah,
approximately 300 plus participants they are listening. One
yeah.
is
Great.
another YouTube channel, another is our presently Google Meets. Sir, the different university peoples like Chaudhry Devilal universities, Badi universities, Grafik Kira universities, and Yuvraj College, Dhan Lakshmi College. So, approximately every institution in India, they are today interact with us for your lecture, sir. So now I hand over mic to the Dr. Dilpreet. He will interact with you. So, Dr. Dilpreet, please.
Good afternoon to everyone, and especially, sir, can you hear me? Irham, sir.
Yes, we can hear you, Dr. Dilpat.
Sir, before starting the presentation, I will briefly introduce about you towards our esteemed delegates. So, our today's speaker is Dr. Irham. Dr. Muhammad Irham is a distinguished Indonesian academic and researcher specializing in pharmacogenomics, drug repurposing, and precision medicine. He currently serves as a lecturer at Faculty of Pharmacy, Universitas Ahmad Dahlan, and during his UG program and master studies at UAD, he scientific article on EMC, medical genomics, and frontiers in clinical immunology. Received multiple awards. So, with these words, I welcome you in the today's session, sir.
Yes, Dr. Dilpat, thank you so much.
Yes,
So,
sir. So, sir, can we start a session, sir?
yeah, of course. Thank you so much. We meet again, Dr. Dilpat.
Or for a good collaboration, sir, with you. Thank you so
Yeah,
much.
of course. Thank you. So, could I start it?
Yes, sir. Please.
Okay, thank you so much, Dr. Dilprit. The honorables at this moment is Professor Ramesh and also Professor Dr. Ikram as well as that Professor Dr. Lafir for very uh, good, sharings between our university actually is between CTU and also Universitas Ahmad Dahlan. So at this time that I would like to present and also little bit share about my research topic of the precision medicines and how's the challenges in the research and clinical implementations. So Dr. Dilpritz, can you see my slide, right?
Yes, sir. Your presentation was visible,
Okay.
sir.
So okay, thank you. And then I will begins. Before begins, I think before I start my presentation, actually Dr. Dilpritz already introduced me, and thanks to Dr. Dilpritz. Actually, I'm right now is a lecturer at the Universitas Ahmad Dahlan, one of the university. Actually, it's the private university in Indonesia, and we are under organizations of Muhammadiyah. Is runs one of the 250 university in Indonesia. And at the same time, I am also the head of scientific publications at Universitas Ahmad Dahlan since 2024, and the young researcher at Silpakon University at Thailand. And what I would like to share to you is about the genomics as well as the pharmacogenomic and also the precision medicines. And one of my research topic right now is about the cancer of genomics and how that we utilizing the genomic informations to drive drug proposing or that we call the drug discovery. So this is my selected publication that I have published regarding of what I would like to share to you for today's events, and mostly that about the precision medicines and also the pharmacogenomic as well as the drug reproposing. So this is my profile in Google Scholars. I have more than the citation is more than 75 7,500 for today's update data, and then for my Scopus profile is I have citation is more than 6,300. So I will begin for my presentation is from this slide. Actually, it's better if we can interact with the our audience. Actually, if you see the slide is I think uh, is there anyone that can guess this slide is or this cartoon is when this cartoon was made? It is there anyone here can guess it? Yeah, I think yeah, this cartoon is was created in 2003. It's a very long period of times. So what does the means? The meaning of this cartoon is the scientists 2003 they hope that when they discover the human genome projects at this time at that times in 2003 and 2004 that they assumes that when the doctor or medical doctor come to the ward in the hospital and they diagnose the patient, they hope that they can easily or they can faster to this diagnose the patients because of the genomic information that it's mean that the laboratorium is more precise so that the doctor can more faster as well as that can more precise to diagnose of the patients. At the same time that also in the future that when the patients came to the pharmacy and then they want to buying some medications for example and then they're asking the pharmacist at that pharmacy in the future I think that the patients can bring the kind of the genetic code they can and then a pharmacist can translate it that kind of the genetic code into the adjusting the dose of the medications. So actually, this is the hope of the pharmacist, and then that is the interchangeable with the future of the pharmacogenomic and as well as the genetic. And I think the slide is showed us that yes, every single of us here like coffee. I don't know in India what kind of the coffee is popular, but in Indonesia we have a lot of the coffee. uh, every area in Indonesia we have a special or we have the own coffee from the specific area. But the mostly I think that we know or we familiar with the espresso, latte, or the cake, the coffee, right? So w- what uh, what the drive or why that the persons or the people like the espresso, why the people other people's like the latte or the cut of the cake of the coffee? The answer is because of the genetic code. So the scientist has have answered this kind of the diversity or difference uh, informations about the why that the persons has different responses to the copy because of the genetic code. So this means that the genetic information is not only help us to identify the medications or to identify the response of the persons for the medications and also the easily to diagnose of the patients for the medical doctor, but also for others purposes. So things about this kind of the that we call the GIES. GIES is the geographical information systems that when we go to anywhere, I think right now we can using and you can utilize it the Google Maps, right? So at the same idea that the also in the medicines right now that we call the omics informations like the genomic, transcriptomic, proteomic, metabolomics, as well as that phenotypes that can be utilized to more detail to know about or precisely to know about that we call the disease, right? Or the same meanings with the phenotype. So this is the idea that in the future that we call the medical doctor, he would like to utilize for this kind of the information. So the multiomics is central to obtaining the detailed big picture of molecular level dynamics, as well as that the previous idea that I showed to is about the geographic information system. So the same also when I think sometimes or someday that or in the futures I can come to your university, Dr. Dilpret, City University, as well as you also come. Welcome to Indonesia and welcome to our university. And for example, if I want to fly or I want to go to your university, right? It's very easy, right? So just go to buy the ticket of the flight, and then we can go directly to the university without any that we call the miss we cannot go to anywhere right now without the flight, right? And also without the Google Maps. So using Google Maps right now is we can easily arrive to your university. So this kind of the idea that also the in the medicine or in the medication system also right now they retrieve this kind of the information's idea to treat it to patient. So one of the widely used or widely utilized right now is the genomic informations as one of the biomarker, or one of the that we call the sometimes is the drug target genes for drug discovery and drug development. So that is for me in this kind of the presentation is I'm very honored to share to you about this topic. Is first the basic of genomic, because we are talking about the human genome, and then the second ones that we call the clinical pharmacogenomic implementations. Because when we are talking about the medicines, I think the pharmacist is the interchangeable uh, person that serve for the medicines, except the medical doctor, right? So the pharmacogenomics that we would like to share to you is about how that the informations of the genetics can influence the response of the patients to the specific drug. So that we would like to share to you is about the clinical pharmacogenomic implementations. So begins from the basic of genomic. When we are talking about the basic of genomic, I think we can begin from the terms of the genomics. So actually, it's the genomic informations or the terms of the genomic is not a very long period of times. I think it comes from one of the journals with the title "New Discipline, New Names," and the journals in the terms of for the newly developments of mapping sequencings, we have adopted the terms of the genomics. So since these journals from the genomic journal in 1987, they published this kind of the article in this kind of the journals. Since at the time that the scientists around the world, they using the terms of the genomics for describing something that it can be passed from the previous generation to the next generation. So what is the genomics? Actually, is the genomic is kind of the plural names of the gene. So the gene is the singular name. It means that the genomic is the field of the genes, or the field of the some kind of the knowledge of or the field of the studying of the genetics or the gene. So genomic is the plural names, but the gene is the singular name. So we are not forget with the Gregor Mendel. Gregor Mendel is one of the first persons that he acknowledged, or he also introduced the terms of the genomics in 1865. But he is not using the genomics as the kind of the abbreviations, but at that time that he introduced to the peoples, he said that there's some things that can be passed from the previous generation that we call it the genomic information. So think about ourselves, right? So our cell is contains or consists with trillions of cells. So inside of cells there is the nucleus, And inside of nucleus, we have every single of us have around 23 chromosomes, and inside of the chromosome there is the DNA. Actually, it's the DNA is the interchangeable terms with the genomic information. So that we are more talking about the DNA, or that we call the genomics in the future of our slides. So DNA is the one of the double that we call the deoxyribonucleic acids. And then after Gregor Mendel's discovered the some things that can be passed from a previous generation that we call the genomic or the genetic. So in the other years, like the Watson and Crick's, two young scientists at the time in 1953, he and Bob Jones are introduced about the DNA is the form of double helix, and the later on that the Nuremberg, Krona, and Holly in 1966, three young scientists at the times, he also completed the discovery of Watson and Crick's by saying that, or by describing that the DNA is contained with the some of the codes, like the adenines always pair with the thymines, and the guanines always pair with the cytosine. So through this kind of the codes in the future, that we can easily to detect the disease, and we can also easily to detect why that the persons or other persons can be other or can be different for responding or respond to the some kind of the medication. So to easily readings the code of the DNA, I think scientists they using kind of the size or length of the DNA. They say that the base pairs, kilo base pair and mega base pair, if the sequencing of the code of the DNA is more than millions of the sequencing. So one of the big projects, and the big projects ever in the world is that we call the Human Genome Project. Uh, at the times from since 1990 to 2003, I think it's around 13 to 14 years that the scientists around the world, they gathers almost more than 50 countries around the world. They discover that's kind of the Human Genome Projects. And then uh, actually right now we are on the post-genomic era that we call the 20 years of big biology. So what does it means? Actually, it's the post-genomic era that we call right now is actually, we can say that the implementation should be in the clinic right now. So it's mean that the all the medical doctor or all the clinicians in the hospital or in the pharmacy should be I think utilize the genomic information. It should be, but different country has different or different regulations. So things to more easily to understand about the genomics. Things about the genomic is like the book, and then inside the book is there is the chapters that equal, or we can imagine it's like the chromosomes. We have around 23 chromosome, right? And then inside the chromosome there is a sentences, and then inside the sentences there is the letter or the word that we call the code of the DNA. So if you are trying to write it down in the books, and then you will spend around 4,200 books. So or what does it means that we call the code of DNA that we have in every single of our bodies is around 6.4 billion letters. This is very huge number of the code of DNA in every single of us. So Dr. Francis Collins is the director of Human Genome Project. He said that he cataloged the 3 billion letter on the human DNA. The data would fill more than 2 million sheet of papers. It means that if you put it on the vertical ways, that you will spend around 168 meter as height. So this is very that's very interesting, because he write it down in 2006 in the language of God of the title of his book. So we are so unique. Actually, the human genome, or that we call the DNA, is very long. So we have around 6.4 billion letters. If someone is an assistant professor here with the ability to type 60 words per minute or 8 hours a day to type it, or to type the DNA code, he will need the more about the 40 years. Is so long period of time, right? So uh, This is the achievements of the genomics since that Human Genome Project was discovered in 2003. Since that the 1990 to 2003, it's span around 13 years. Since the Gregor Mendels at the 1853 discovered the something that he said that the law or some things that can be passed or from the previous generation to the next generations that we call genetic. And then later on that the Watson and Crick discovered the double helix of DNA, and then later on also in 1966 that we call the Niramba, Purana, and Holly. Here they three of them are discovered about the code of the DNA, and then later on that there's the human gene biobanks and also the Human Genome Project in 1990 to 2003, and then since 2004 there's a lot of the animals models and also the human can discover the code of the DNA inside of his body. So along with that, also in the every years right now that we can see around the world that the developed country and also developing countries, the building the DNA that we call the genes banks or that we call the biobank. So including our countries right now, our governments trying to build the biobanks and then collaborations with China to build, because of in Indonesia we have that we call races, more than 300 races. So this is a very huge number, and then we needing the biobank to discover a lot of information about the genetics. So this is the information about the biobanks right now that we can see. But this is in 2000 or almost 4 to 5 years ago. But I don't know about the updated information right now. Actually, I think it's more than this kind of the slide about the biobanks in around the world. I think India also has own the biobank, right? So including our countries trying to collaborate with the China to build the specific biobank. So when we are trying to see the Human Genome Project right now, so when we can divide it into three parts. First is that we call the pre-genomic era, and then the second one that we call the post-genomic era, and the third one that application of genomic era. So what's the pre-genomic era? The pre-genomic era that we call from 1990 to 2003 is the understanding the structure of genomes and understanding the biology of genomes. And then for the second one that we call the post-genomic era is the understanding the biology of disease. And for application of genomic era, actually is the interchangeable terms that we call the genomic medicines, and hopefully that improving the effectiveness of healthcare. That meaning that is the implementation of the genomics should be in hospital or in the pharmacy or that we call the in the clinical system. So if we see right now the implementation of genomics, I think it's diverse, but the specific area that we call the hot area of genomic medicines versus the cancer of genomics. Why the cancer? Because the cancer is one of the diseases not only to identify the cause that we call the biomarkers is very hard, right? So not only too hard to diagnose of the patients, but also to treat it because of the mostly medications that treated to the cancer patients is troubles in the resistance, right? So that we call the resistance of the medication. So second one is the pharmacogenomics. The pharmacogenomic is the uh, combinations between two kind of the field like the pharmacology and the genomics. So that we call the pharmacogenomic in the or if we would like to simplify, it's like the response of the drug is the influenced by the genomic information. So the third one is that the rare genetic disease. Rare genetic disease is also uh, very hard to diagnose to the patients and also the treating. So hopefully that the genomic informations can more easily to diagnose the patients as well as to treat it the that kind of the disease. So one is the prenatal genomic testings, and also the fifth one is that we call the discover or drug discovery. So this is actually is nowadays I'm going to do, and also I'm mostly of my project right now is on the genomic driven drug discovery, meaning that we can utilize the genetic information to identify the candidate of drug of the some kind of the disease. So I would like to jump to the second of my presentation about the how the clinical pharmacogenomic implement in the area of the hospital or in the pharmacy. So when we are begins from the pharmacogenomic, I think we already heard about the pharmacogenetic, right? So actually, it's the what differentiates between the pharmacogenetics and pharmacogenomics. Actually, these two kind of the term is the interchangeable terms. So what is difference? The difference is the pharmacogenomics that we call the is the plural name of the medication is the response is has been influenced by the many genes, but for the pharmacogenetic genetic is the single genes. So it's mean that pharmacogenomic is the plural names, but the pharmacogenetic is the singular name. So uh, relevance or that we call the related the terms that widely used is like the pharmacogenomics, interchangeable names with the personalized medicines, and also the precision medicines, and also the individualized medicine. I think this kind of terms is interchangeable terms between the pharmacogenetic as well as the pharmacogenomic. So one of the implementation of the genetics is that we call the single nucleotide polymorphism. So single nucleotide polymorphism is uh, very pivotal roles in our body. So that we call the genetic variations. The genetic variation is synonym with the polymorphisms or single nucleotide polymorphism, the flips in the single of the base pairs. So we are actually very similar, but little bit different, meaning that's what almost 99.9% we are similar to code of our DNA codes, and then the difference is almost 0.001%. It's meaning that the tiny difference in our code of DNA can be influenced or can be affected with the associated with the disease as well as that uh, the track response. So this is the answer why sometimes that the medicines B might be better than medicines A for every single of us, or medicines A better than medicine B. So this is the answer is because of the single nucleotide polymorphisms. Why that? Because the single nucleotide polymorphisms, or that we call the genetic variants, is the very uh, very common in our body, and almost that the scientists around the world said that almost in every thousands of the sequence of code of the DNA, there is one of the SNPs, or there is one genetic variations. So the consequence of variation is diverse, but right now, if we see from the publications, many of publication has been published, they discover or they inform us about the uh, many kind of the consequence, like the drug transporter and also the drugs receptor sensitivity. Some of the persons has receptor sensitivity, and another one is there is no sensitivity to the receptor of the medication, and then the one is the response to the non-response. Why that this person is non-response? Why that the person is more faster to respond? Because of the answer is because of the polymorphisms. And the other one is the adverse drug reactions or adverse drug event. I think very common right now. One of that is the abacavir. Abacavir is one of the medicines for HIV that has been influenced by one of the genetic variants, like we call the HLA-B star 57. So another one is the disease susceptibility, and then also drug metabolism. Also, drug metabolism also has been widely informed through the many publications. They said that the medications influence the metabolism of the medications or medicines has been influenced by the single nucleotide polymorphisms. So Jago is the in Indonesia. This is actually is the documenter documenters or film documenters in Indonesia. That's I think is very popular. This kind of this race. Uh, uh, Jago meaning is like the master. What they describe in this slide is Rohani is the Mr. Rohani is come from one of side of our countries that we call Sulawesi. Daily activity of Rohani, Mr. Rohani, his age is more than 80 years old. Daily activity, he always, as the fisherman, actually it's the fishermen. And then the unique, this is the unique person. This is not only unique persons but also the race, unique race, because of the he is with his family and another family with the, from the Bajau races in Sulawesi. He always find the fish underwater is without the appropriate equipments. It's meaning that the most important things from the Mr. Rohani, he can survive underwater with a single breath more than 10 minutes. So we can imagine, right? So more than normal people. I think normal people can survive underwater more than 1 to 2 minutes, I think. But for Mr. Rohani, he can survive underwater more than 10 minutes. What did the answer? The answer is because of the genetic. So I think this is the Rohani is from Suku Bajo, is kind of the races from one of the country, one of the part of Indonesia that we call the Central Sulawesi. So the answer is because of the genetic code. So the genetic code is that we call PDA10A, and then second one is BTKRP2. Indirectly, that can more has the pivotal roles in the increased spinning size. So in direct terms, that we call the this kind of increased spin size in the Bajau races can providing them with larger reservoirs of oxygenated red blood cells. So this is the answer why that the race of the Bajau in one of the part of Indonesia, he can or they can survive underwater is more than 10 minutes. So because of the genetic code, the answer. So I think we are right now is on the one drug fits all approach. So what does the mean of the one drug fits all approach? One drug fits all approach meaning that one dose for all. One dose for all. See an example here is 100 milligrams. This is no difference between one person to another person. But when we would like to implement the personalized medicine or precision medicines or pharmacogenomics, that we can implement it, we can adjust it to the dose of the patients based on the metabolisms of the patient. Like the patients with poor metabolism should be decreasing the dose, as well as that for the patients with more rapid metabolism should be increasing the dose, so that the patients can be achieve this sustained state or steady state of the medication in his blood, right? So uh, we move to the precision medicine. So what does the mean of the precision medicine? Actually, precision medicine terms come from Mr. Obama. Mr. Obama is, we know that the former president in at the United States, right? In 2015, he is the president at the United States at the time that he launched one of the concept of precision medicine initiative to bring us closer to curing disease like cancer and diabetes mellitus, to give all of us access to the personalized information. So since this kind of the concept introduced by Barack Obama at the time in 2015, almost all the scientists around the world, they're using this kind of terms, personalized medicine or precision medicine. So in the previous terms, I think the personalized medicines or that we call individualized medicine is the more popular terms rather than the precision medicine. But since that Obama introduced this kind of terms, that more and more scientists around the world, since that the more publication has been published, they using the terms of precision medicine. So the concept of precision medicine is the combined between the variability of the genes, environmental factor, as well as the lifestyle factors to treat or prevent the disease to the patients. So this is the concept of precision medicine by using the combined between the variability of the genes that we call this variations or single nucleotide polymorphisms, and also the environmental factor and also the lifestyle. So what is the aim of the precision medicines? Actually, the precision medicines aim with earlier diagnostic that we call better quality treatments and also the better disease classification. And the second one is the widening the possibilities of prevention disease. And the third one is prediction of outcome, cost benefit, and also the cost effectiveness of the medications. But even that the precision medicine have, I think, so many informations and also uh, the possibility to implement in many countries, but especially in our countries, there is a lot of challenges, especially in the evidence generations and also the data sharings between the one countries to another countries, and also the incorporating personalized medicine into the clinical care also is a barrier here. And then the fourth one is the diagnostics, or that we call drug discovery, and also the economic barriers of precision medicines, because of not health insurance can cover this kind of the medication. So for fifth one is the participant engagements and also the regulatory challenges. So I think this is a part, is a small part of the challenges. I think the other country has their own uh, and the challenges, right? So to implement the precision medicine. So but another from the challenges also that we can also discuss about the benefit of pharmacogenomic right now. So the benefits, I think, can decrease in admission of the patients, as well as that can suppress the expense of the from the patients. And then another one is we can uh, support or we can also the increasing the safety, efficacy, and compliance or adherence to the patients, because of the since that patients would like to treating by some specifics of medication, they should to test the genetic first. Then they can avoiding they can avoid the adverse events of the medication, and also of course that can be enhanced the safety, efficacy, and also the compliance to the patient. So in the future, I think the genetic code or uh, the testing of genetic code will be very popular in the future, in the futures, and then I think it's like the dreams come true. Why I'm saying like this, because if we see several decades ago, for example, in 2001, 2002, 2003, and since that Human Genome Project discovered since 1990 to 2003, I think it's more than hundreds millions dollars they should spend for one person to identify the genetic codes. But right now, in more than 2000s, I think above 2000s, people can testing the genetic codes with less than 1,000. So it's meaning that in the future, it should be, I think it should be, as I said, as aforementioned it before, that the dreams come true. Every person can testing the genetics for testing his own genetics, so they can easily to identify the diagnose of the disease, as well as that to identify the future of his disease, and also for the future of his for treatment. So if we would like to more details learns about the pharmacogenomic, right now is the lot of the websites, lot of the own groups, and also the pharmacogenetic groups and consortiums that they provided a lot of information about this kind of concept of pharmacogenetics. But in here that I would like to highlight several popular website that has been widely used for identifying the pharmacogenetic information. First is that we call the CP guidelines. CP guideline is the Clinical Pharmacogenetic Implementation Consortiums, and then second one is DPYG, is Dutch for Pharmacogenetic Working Groups, and third one is the PAMGB. PAMGB is the Pharmacogenomic Knowledge Base, and then one is the PAMFAR, Pharmacogenes Variations Consortiums, and the fifth one is the PGRN, is like that we call the Pharmacogenomic Research Area. But mostly amongst this fifth website, I think the most popular one is the PAMGKB, and also DPS CPIG. CPIC is the two website database is mostly used in the world right now. So if we would like to more detail to learn about pharmacogenomic, I think you can spend your time to identify it or to learn more details through this website. And then this is also right now, it's not only the website of pharmacogenomic that I can provide it here. And if you would like to more details to learn about the bioinformatics, because right now is the bioinformatics is, we can say that the interchangeable field between the genomic information and also the pharmacogenomics, like several of database right here. You can easily or you can identify by yourself, like the Haplorec, Ensembles, Nexus, CB Portal, Encode, TrackBank, JIB, clinicaltrial.gov, GTEx Portal, CMapStones, GWAS, PWAS, Study, and others kind of the website database that this is actually is my I summarize this kind of slide is for you. If you would like to, because we don't have a lot of time, right? So I think it's because of shorter times, and then I summarize this kind of website that you can uh, easily to find in your homes, or this is as the take home message. So this is a lot of my publication is mostly almost about the genomic and also the pharmacogenomic and also the bioinformatics. This is for COVID-19, and another one is for the hepatitis B and also the hepatocellular carcinoma. And also this is the genomic drug discovery. One of the my publication has been published in the pharmacological research by utilizing the genetic informations. And then another one is the hepatitis B, chronic hepatitis B, and then another one is the tuberculosis, because one of the problem in our country in Indonesia is still in tuberculosis treatment, and we are trying to find the right medication for tuberculosis by utilizing the genetic informations. Another one is for asthma disease, and also the uh, another one is for antidepressants that we call the depression's disease, and then that antidepressant is emergence to identify the special or specific candidate of drug target genes. And another one is the currently that we also with our teams we published about the hemorrhoid treatments, because in Indonesia is still a problem hemorrhoid. And another one is diabetes mellitus, complicated with the diabetic foot ulcer, and also for diabetes mellitus, right? Especially for type 1. So we identify the candidate of the specific genes. Another one is the multiple myasthenia gravis. This is my from my master students. Another one is for liver cancer, and also for systematic lupus erythematosus or SLE. And I would like to, I think, to end up my presentations by uh, providing you the information that the pharmacist is not only role in the pharmacokinetic and also pharmacodynamics uh, uh, informations, but also for educating and also the counselings of the pharmacogenomic in the future. And then, of course, this is our job. This is our duty in the future, and we should to start right now to learn about pharmacogenomic. And then, of course, that in the future, the pharmacist, also the other medical doctor also can interpret the clinical pharmacogenetic test result to the patients. So pharmacogenomics, even that we have many opportunities to implement, we still have many constraints in the clinical practice right now. It's like the facilities and also the cost, because in our country, especially in Indonesia, the pharmacogenomic testing is not covered by our national insurance, BPJS, the name of Indonesia. And then, so for one is the ethical issue, because of the genetic information is the own uh, is very secret, right, for every single of us. So this is the ethical issue should be clear in the future. And then the other one is about the variation in drug response between individual are highly variable, because in Indonesia we still have more than 300 phases. So I don't know in India. I think it should be more. I think it's more possible to implement it. And another one is the role of clinical pharmacists in the future. I think it should be interpret of the gene result, and then drug selections, recommended drug dosage, and also the collaborations with clinicians to make treatment protocol and provide education and counseling and calculating drug cost or drug discovery and drug development. So this is the special or specific publication that they're describing about the pharmacist's roles in the pharmacogenomic implementation. So I would like to conclude this separate part of my presentations by saying that the post-genomic era is the that we call the genomic medicines we are right now is should be implemented. I think even that for today's we cannot see in some part they cannot implement, but in the future should be. And then second one is pharmacogenomic toward precision medicine lead to better understanding the interactions between drugs and also the organism. And pharmacogenomic is able to optimize drug therapy and also implement on pharmacogenomic in clinical practice is feasible and effective, although it is still a long way to implement in clinic. And another one is pharmacists are at the forefront of pharmacogenomic, and of course, the precision medicine is global agenda, and we need to participate in international consortiums. So this is my one of the books that we already published about the what I'm talking today about the precision medicine. So thank you very much for the committee member, especially Dr. Dilpritz, for giving me the opportunity, for giving me the times to share our knowledge, to share our study in Indonesia, in your university, in City University. So actually, our university is located in Yogyakarta. If you are welcome to Indonesia, I think Yogyakarta is the central of the university, and the most university in Indonesia is was located in Yogyakarta. And then the biggest temple in the world is in Indonesia is was located in Yogyakarta. There is two temple that we call the Borobudur and also Prambanan. So welcome to visit our city. And then another one, we have a lot of the part of Indonesia, and we are right now is we have around 4,000 university in Indonesia, and then we some of part is very beautiful. I think so you can, as you can see in the slide, and then of course, Jakarta is our central capital in Indonesia, and East Java you can see here, and then Bali. I think here is more popular Bali, right? So Bali is one of the tourism destinations in the world, and then Papua New Guinea is the east part of Indonesia, but very beautiful. And then another one is west part is that we call Sumatra. They have a specific area that very famous there, and also the Sulawesi. And I would like to thanks and then welcome to Indonesia for the future. And thank you very much, Dr. Dilprat, and send it back to you.
Thank you, Dr. Muhammad. So that is a nice presentation. So there is, I think, only one questions, that is, no. So what are the biggest obstacles healthcare system face when integrating precision medicine into routine clinical practice? That is questions asked by Dr. Shikha.
Okay, thank you so much, Dr. Dilbert. I think the obstacles right now is mostly about the cost. Mostly about the cost, Dr. Dilbert, and the cost, not only the cost, but some countries also about the data, and also, for example, in our country, in Indonesia, our government doesn't has an own biobank. So how could we that as the reference? The biobank is the what as the reference, right? So how could we identify the specific genes that uh, specific associated with the disease, or specifics can be implemented with some kind of the disease if we don't have the biobank? So for some of developing countries, it's because of data sharings, and then or that we call the understand the biology of disease. But another one also the cost. The cost is still huge or very expensive. So this is the in my opinion is this two kind of the information as the most challenges or most obstacle to implement in clinics. That's my answer, Dr. Dilprit.
Yeah, you are absolutely right, doctor. So another question that is asked by Dr. Jaswindra. She asks,
In the EI website to identify the specific candidate drug target genes for specific the disease. So I think uh, very fast to see right now the implement or that we call the integrates between the artificial intelligence and also the leveraging the genomic informations to identify the or that we call drug repurposing. Drug repurposing is like the using old drugs or utilizing old drug for new indication. So mostly that I see right now the artificial intelligence can be leveraged for that purpose. That's Dr. Vikram, my answer.
Yeah, it's so one question by Dr. Dilpreet. Please, Dr. Dilpreet, ask your question.
Sir, I have one question that what particular factors that we need to consider for optimizing drug therapy in pharmacogenomics?
Yeah. So you mean that the fact, pardon, Dr. Dilprets, could you more specific your questions?
Sir, actually, my question is that you said that there is a role of optimizing drug therapy in pharmacogenomics. So, what particular factors that we need to consider? Any
Yeah, of
specific
course.
factors?
Yes, thank you for your very excellent question, Dr. Dilprets. Of course, that the genetics informations or we call pharmacogenomic is one of the factors, right? In the influence the response of the persons for the drugs. There is the environmental factor, lifestyle factors, and also the all patients or we call age of the patients. So the genetic information is one of the specific factors that more molecular factors that has been widely informed us from many publications. But right now is the still challenges is about the that I call aforementioned it's about the disease associated genes. Some of the genes, for example, is specific in the some disease in some countries, but not for other countries. So this is also still debatable. So I think it's from CPIC guideline and also PAM-GKB, because PAM-GKB and also CPIC guideline right now is the integrated with the FDA. So right now is every FDA approved the medication, especially for cancer therapy. They should do they're asking the company or industry to provide the pharmacogenomic information. So one of the widely implement is the that we call abacavir. Abacavir is one of even in that in our country in Indonesia, abacavir is the one of the standard treatments for HIV. But the problem is in the hypersensitivities like the Stephen Johnson syndrome. So before patients given the abacavir for specific HIV patients, they should do testing the genetic first, and then they consider whether the patient's carriers of HLA-B star 57. If the patient has an own specific variations in HLA-B, they should to avoid to given the abacavir. So that we call Dr. Dilprets the factors of genetics. One of the I think small factors, and another one is the environmental factors. Of course, the age of the patient's lifestyle is very that we call influence in the response of the medications. That my short answer, Dr. Dirpit.
Thank you. And Doctor, there is one question by Shikha, Dr. Shikha. How can hospital and clinical without high-end genomic lab participate in precision medicine initiatives?
Yeah, thank you. This is excellent questions, I think, Dr. Vikrams. Yes, of course, that the how can that the hospital can be participate in this kind of in the future? I think for the precision medicine, I think we can see right now is the almost that the almost all countries right now in especially in developed country they have since like the electronic medical record, right? So the electronic medical records more I think in the future should be more faster to implement the precision medicine because of the information that put in the electronic medical record is of course that very fast to collect, and also of course that very fast to integrate with between the information in the electronic medical record and also the from the other database. So this is, I think, is more that we call the more popular that hospital can start to or can contribute to the implement of precision medicine. But the problems, Dr. Vikram, right now is the ethical issue. Ethical issue. So some kind of the insurance company, they want to know about the specific genetic of the some person. So this is also the ethical issue. Even in developed countries, is they are trying to protect the patients from this kind of the issue. That Dr. Shika, I can answer, I think.
Sir, it's a great answer towards you, sir. And there is one last question that asked by our colleague, that how can clinician be better trained or supported to interpret and apply precision medicine data in real-time
Yes.
patient care?
Thank you so much, Dr. Kumar, Dr. Akshay Kumar, as the assistant professor at City University. Yes, that's totally right. So how could we start it? I think if, every single of us, we can contribute it, right? So we are trying, if we are professor, if we are lecturer in some of university, I think we can starting from the curriculum. Curriculum is like the in our countries right now, the pharmacists in the bachelor degree, uh, the pharmacogenomic is still elective, but for a pharmacist, because we are the bachelor and the professional programs, we are different programs. But from the professional program and also master program, this the how would we say that, like the um, bioinformatics and also pharmacogenomic and precision medicines lecture, they are mandatory to take. So this is the answer, how we could be trying it, because of the some of the pharmacists right now, they are totally doesn't know about the pharmacogenomic. But why? Because they don't have the lecture in his study, during his study from the bachelor until his professional program. So I think this that we can starting from now is, if we are lecturer at the university, I think we can start it from the curricula at our university, whether it should be elected or whether should be mandatory. But for in our country, I think in the future, I think in the bachelor degree, they should to be mandatory, because of the in the future, I think we are facing the real word about the how that precision medicine should be implemented more easily to the hospital, and of course that we can educate the other medical or nursings or medical doctor or other clinicians, and of course that counseling to our patients, so that we can trying to educate as Dr. Askay Kumar. And another one, I think, is that for if we would like to if you would like to join the training about the pharmacogenomic, I think some of the specific hospital in our country, I think is in our central, in our capital city, the one of the hospital that we call the Ciptomangunkusumo is one of the biggest hospital in the our capital city. They are educate some of the pharmacists to educate for education of the pharmacogenomic in the clinic. That my answer, Dr. Kumar.
Thank you very much, sir, for the great answer. So now we will like to conclude the session. First of all, I would like to thank you, Dr. Irham, for the wonderful lecture.
Yes.
Sir, session delivered by sir proved to be highly impactful and intellectually enriching. So the discussion provide advanced research with practical application, especially in precision medicine and genomics. So I would like to invite Professor V. Vikram Sir, Principal, SOPS City University for vote of thanks. Sir, please.
So again, thank you, sir. It was a
Thank
wonderful
you.
interaction because in India it's our lunch time, but we have skipped our lunch
Thank you.
only to interact with you. And
Thank you so
my
much.
all faculty members, students, even we have telecast your lecture on YouTube, sir. So I will share the all documents to you.
Yes.
So with folding of my hand, thank you from the India. You have done excellent work, and I really appreciate your work. Whenever I see your slides, you are working on tuberculosis, you are working in different disease. It's incredible. So I request to the Dr. Dilpreet, please show a memento of love from our side, a certificate of appreciation.
Thank you so much, Professor Vikram, for the opportunity.
So even though in future we can collaborate,
Of
because
course,
just
Professor
like
Vikram.
Tokyo University, I visit there for collaborative work. But in future, in your country, in your lab, India and your countries collaborative do research work in the field of medicines, in the fields of disease, so that it's a betterment for the humanities. So we will love to listen from your side in future collaborative work.
Thank you so much once again, Professor Vikram, for the opportunity.
Thank you. So it's a sir award of honors from our side, and accept our sir little bit gratitudes for you.
Thank you once again.
And thank you all the participants who join us today's session. And the next session is on 26th, and we will, sir, even though your students has joined now
Yeah.
this session, 4 to 5 students of yours
Yeah.
and
Some of my students joining this
yeah,
sessions. Thank you, Dr. Vikram.
yeah,
Hopefully we can meet together.
yeah, definitely, sir.
Thank
Thank,
you.
thank you.
Thank you, Dr.
Thank you very much.
Dilpreet. Thank you. Bye.
Sir, just leave the call.